Okay. Perfect. I'm going to log in a hot second.
No worries.
You know, I got to like
Do what you need
tell
to do.
everyone to join. Get on this call.
Make this into a shape like a wall.
And then we're back to like 50 years ago.
Right, yeah.
I remember those. That
Oh,
wasn't
yeah,
even that
yeah,
long
yeah.
ago.
No, it wasn't. Because like all of my physics classes were like
Transparency
that.
and wands were like,
Flapping the board.
I'm going to do this. I'm going to buy one of these. And then I'm going to buy a wand, put into a wand, and then I'll bring to some Janelia event to do the presentation.
I'll do that. I'll wheel a slide projector out.
Let's do that.
I feel like the slapping is something we've lost.
It
Right?
is.
I mean, back in my day, you could catch people's attention. On slide 16, whack. Error bars are great.
Can we coordinate and then do this on the same like progress block?
There we go.
We could print out transparencies and write on them on Sharpie.
Yeah.
And then smash the board with them and really bringing back the good old days. I like half did this at a talk at UCSD like over the summer, and I insisted on doing a whiteboard. The whole thing I just like
Yeah.
wrote it out.
You're really old school.
I like old school.
I think we should go for it.
Okay. Great. Good morning, everybody. We're starting the last day of this wonderful conference. This will be a session on tissue biology tools. And we'll be kicked off with Jim Wells from the University of California, San Francisco, who will tell us about new tools to probe the surfaceome.
There you go.
There we go.
All right.
Good morning.
Wow. Okay, thank you very much. And thanks to Jennifer and Ron for inviting me and Janine organizing. This has been a great meeting. This is sort of I don't come to a lot of neurobiology meetings. It's not my field. Normally I would be at a protein engineering, drug discovery, cell biology type meeting, but I've learned a lot, although I feel a little bit like a zebrafish out of water, if you can imagine. My lab studies this, which is a cell with a bunch of membrane proteins on it. We call that the surfaceome, the surface proteome. And it's really critical in biology. Obviously, all the communication with the outside world is done through these proteins. Nutrient import, metabolite import, et cetera, is done through them. Communication with hormones and receptors and the immune system and adhesion and all of those functions. And it represents about 20% of the proteome. So it's a very significant genetic commitment to the surfaceome. It is also the home of most drugs. Half of the small molecule drugs engage this compartment, and virtually all of the biologics engage this compartment. And my lab studies how the surfaceome changes in health and disease, because in disease, new proteins often appear or change in their expression levels or their complexes or post-translational modifications that can be targeted with antibodies, which is another big part of what my lab does, antibody engineering. So I wanted to first tell you sort of the levels that we kind of probe the cancer surfaceome or the immunological surfaceome or cellular surfaceomes. One of our favorite methods is to be looking at the what's up team, which is how do protein levels change when oncogenes go into cells or they get inflamed or something like that. And we've developed some, in addition to established methods like hydrazide, which pulls out membrane proteins, and you can then assay them by determine what they are by mass spectrometry. We've also developed a newer approach to this using an HRP that's been tethered to the cell membrane, wheat germ agglutinin HRP. Actually, this is commercially available. It's a very democratic reagent. You can get it yourself. If you add it to cells, it will just start biotinylating in the presence of biotin-tyramide membrane proteins. And this tool here allows you to get about 5 to 10 times fewer cells in the proteomics process. So it's very useful. We're using it most recently for microglial cells and things like that to find out how their surfaceomes change from an inflamed to normal situation. Another very important process that happens on the cell surface in health and disease is proteolysis. There's 500 proteases in the human genome. Most of them have their active sites facing outside, and they're highly regulated, and they are induced to be active in certain disease situations, especially cancer, for instance. And so we've developed a cell-tethered peptide ligase called subtle ligase that will allow us to do what's called N-terminomics, tag the N-termini of proteins. And of course, N-termini are created during proteolysis. And what I'm going to tell you about today is these have recently been published. So I thought what I'd talk about today is work that's none of it's published. And that's an area that we're also very much interested in, which is the what's in the neighborhood team, which is what are the protein complexes that change during oncogenesis and during signaling and things like this. And we utilize a new technology that was first described by the Macmillan and Osland Nadehe groups at Princeton and Merck, respectively, which uses highly reactive carbines to find out what's next to each other on the cell surface. And this work has been done by postdoc in the lab Thomas Bartholow and a graduate student, Paul Burrows, and in collaboration with a group at Merck who's supplied us reagents that we've used for this technology. So when we think about membrane proteins, it's good to just get kind of a length scale understanding of what's there. So these are kind of just a small rogues gallery of protein complexes. And you can kind of see the maximum dimensions of these. Typically 100 to 200 angstroms is kind of the distance from stem to stern of a membrane protein. And you want something, a proximity method that would be respective of the resolution of the problem that you want to solve. And so it's kind of in this range of resolution that we'd be looking for proximity labelers. And so there's really a sweet spot for proximity labeling, and I show this to show sort of this nautical analogy here. That, imagine we have a protein complex, this cargo ship with containers on it. We want the proximity method to identify what's on the ship, but we don't want it to see ships in the night or bystanders. So we don't want it to be too long. We want it to be, and we don't want it to be too short either. Otherwise, if we just stick it on one of these containers, we're just going to detect an immediate container, not the whole complex. Now, I show this as a static view. In fact, these things are very, very dynamic, as you can imagine. Things are moving in, things are moving out. So we also want something that's going to be fast enough to detect sort of a time average of what's there. So what proximity methods are available? There's chemical cross-linking, which has been popularized by the Huang and Burlingame labs. This uses these amine reactive cross-linkers that have a dimension of about 12 angstroms or so between lysines. So if you add these cross-linkers, in order to get a signal, you have to have adventitiously 2 lysines within that distance of each other. So it's very high resolution. But the complexity of this, the mass spec from these reactions is such that you can really only use it, and has been used really effectively on purified protein complexes, because you get half reactions. There are amines everywhere on the cell surface. You can imagine that you just get, it's a really complicated spectrum to try to deconvolute doing that on cells. So it's pretty much limited to purified protein complexes. On the other hand, there's Apex, which I think most of you are aware. This is a really effective way of identifying, for instance, proteins that are sitting on an organelle or across organelles. This has been popularized by Alice Ting's group, which has done beautiful work here to show that with a biotin phenol, you can generate these free radicals that will tag proteins on the cell surface. The resolution of this, recently measured in the Macmillan group, though, is around 3,000 angstroms. Okay, that's sort of the distance that it sprays these radicals. And so that's going to be, you're going to get a lot of false positives that way because you get a lot of bystanders. So here it is again. These hydroxy radicals have half-lives on the order of about a millisecond. And this new method that was developed by the group at Merck and Princeton uses, instead of aryl hydroxyls, it uses azurines to generate carbines, and these react incredibly fast. So you get much more precise labeling. And using their STED microscopy, for instance, they could show that whereas Apex would have something on the order of 3,000 angstrom resolution, these had gotten down to about 50 angstroms resolution, which is really pretty cool. You're starting to get down into the, I meant to say 500 angstroms resolution, 50 nanometers. Now, the way that it works, the way that they had sort of developed this, is that they have a primary antibody that targets a protein of interest, for instance, CD45. And then they have a secondary antibody that's labeled with their emitter. This is an iridium catalyst that will, when you shine light on it, generates a triplet state that then, if there are carbines diazurines nearby, it generates carbines, and so you get localized labeling. Shown here. So the diazurine gets converted to a carbine, and that labels the proximal proteins. But as you can see here, the labels, the emitters are put already 170 angstroms away from the primary target. So you lose a lot of that sort of resolution just in the placement on a secondary antibody. Though it is a democratic reagent, you can use it on a lot of different antibodies as a secondary. Moreover, they used amine labeling to put these emitters on, and they typically have 6 to 8 lysines that are modified, and that labeling is random. So there's 50 lysines or so on an antibody. So you can imagine 6 to 8 sites. The heterogeneity of that is going to blur the resolution. And so we wondered, what is the resolution from a single site emitter? Will single site emitters on primary antibodies, in other words, if you put it up here, would you increase the resolution, and what would be the results that you would get? So to create single site labels on an antibody, we chose our favorite scaffold, which we use for all of our antibody engineering studies, which is derived from trastuzumab, the 6th top selling drug for called Herceptin by Genentech for breast cancer. So we take this Fab and we use a chemistry which allows us to singly modify this Fab any place we want, and that's using a methionine labeled labeling technology that was developed in Chris Chang and Dean Toast's lab, and this oxazurdine, which will then create a sulfimide and allow us to then click on basically whatever we want. And we applied this systematically to scan about 150 sites on this antibody with methionines to find out the sites that are, very high yielding, very stable, very, don't impact affinity, et cetera. And so then we're able to place these emitters, which finally look like this, with the iridium catalyst on one end, tethered to the protein at this end. It's about a 30 angstrom leash. That'll be important. And the Fab itself is about 65 angstroms from top to bottom. So the first thing we wanted to do was just study self-labeling. In other words, if we put this on the Fab and then let it spray the carbines, where does it label the Fab, and what's the distance relationship of where the emitter is to where the labeling is happening? And that's, here we put it on this residue, serine 75 converted to a methionine, that leash put on it. And these are the spots that we label. We label about 100 sites on the antibody. And as I'll show you, it's very even in what it labels. It's not just focused on tyrosine or lysine as those other methods are. This will label anything. We put it on 8 other sites as well and studied their self-labeling. And what we find is that each labeler gives a different spectrum of labels. And if you look at these in aggregate, and I'm just going to go through them quickly because I can see I'm running out of time. You get this Gaussian distribution that's centered right around 30 angstroms. This is the length of the fab. So the labeling actually extends all the way through the fab and then beyond. It's even in what it labels. You can see these amino acids, 20 amino acids here. These two sites, the isoleucine and methionine sites, were buried so that we don't see them getting labeled. But these other sites, it's very even in what it labels. So it sprays everywhere, inserting into CH and CC bonds and the like. Now, if we add then HER2, which is the target of Herceptin, for instance, from this one emitter site, R66, which is right up near the fabs, it's right near these are the sites that we see labeled. If we then compile all 8 emitter sites together, looking at what gets labeled on Herceptin, you get, this is the self-labeling profile. This is the labeling on HER2. So you can see that at a distance, you get less labeling, but plenty of labels to identify things. And we get this, we also get from this data kind of the maximal distance that we can label from one emitter site. It's about 120 angstroms. So then we wanted to start using this on cells and see what happens. And so what we did was to take our fab emitter fabs and bind them to HER2 on cells, and then ask the question, what visits HER2 when we start spraying? And we compared that with a secondary antibody that had been demonstrated from the previous work that I showed you about. And we wanted to compare the number of proteins labeled, the confidence in them, and things like that. And for this work, they used as a control, compared everything to a non-primary added antibody. In other words, just the secondary. It's not really a fair comparison because the secondary alone in solution would just spray like a helicopter trying to mow a lawn as opposed to putting it right on the membrane. So what we also did was develop a fatty acid labeling reagent, which is basically putting a lipid on this iridium and then label from there. And when we did that, we got, this is what we got from just one emitter site, this volcano plot. And using a very stringent false discovery rate, and with 3 biological replicates, 2 technical replicates, we get about 20 or so proteins that are found in this highly enriched, significant space relative to the nonspecific control. And we did this from the secondary. We actually got far fewer in this highly significant range. Of course, HER2 comes out as one of the top hits in both of these. In the Fab 66, when we put it right on the Fab, we got many more enriched proteins at a very high level. There were 4 proteins of the 5 over here were in common, so we captured basically with this all that were found in the secondary antibody. And when we look at all 8 emitters sites, we get these volcano plots here. I'm not going to take you through all of that. We find that each emitter site gives on average, somewhere between 20 and 40 hits that are of high significance, more than 6-fold. So this isn't, I don't believe any of these are a static complex. I think this is reflecting dynamic things that are moving in and out of HER2. Now, the HER2 interface is about as well known as Times Square, or thought to be. But in fact, these are a number of really highly significant hits that we find at this neighborhood, including a phosphatase that we believe is probably dephosphorylating the receptor once it's been activated, a protease that's known to be linked to HER2 relapse, TGFR1, which is really interesting, and a number of other things that are highly correlated with breast cancer, but never associated with HER2. So that was very interesting. So where we're going with this is to generalize this to many other therapeutically interesting protein neighborhoods. You know, HER2, the 6th best-selling drug, that neighborhood still has a lot to mine from it. What will we find? If we find them, can we build bispecific antibodies, making them even more selective, et cetera? to deorphanize ligands, this is something that we're really excited about because we find a number of things are shed from the cell membrane, and what do they rebind? Do they bind to other things? Using this approach, we can find that out. And then we're also using this with the idea that we can use these emitters almost like a multiplex FRET to define structures, protein structures. So with that, I will stop and thank all the folks from my lab. These people in bold are people who are extending this technology. My collaborator at UCSF, Andre Sali, the Merck group, Rob Osland and Nihi Fadehi, who provided the reagents, and Bernard and Paul for managing the collaboration, and these folks for their funding. So thank you so much. Happy to take questions.
All right.
Awesome talk. I read that Macmillan paper, and I was like hoping that you would make the Lamborghini version of that. So I'm wondering, how do you think that iridium catalyst and also the linkage you chose would perform in more sort of complex tissue environments?
I think it should perform just as it did on cells in terms of the chemistry that's there, and so we're actually starting to apply it to label tissue slices and things like that with these very specific DET probes, and we'll find out. I think it will perform in that situation.
Any world in which you can get this to work intracellularly?
Well, actually, the Macmillan group has shown that you can actually deorphanize small molecules with cell-penetrant versions of these. So, yeah.
Oh, I have to make a choice.
That was really interesting. I have a couple questions, but the one's technical, just so I understand. When you were talking about all of the different, that there were different emitter sites and that you were showing that there was potentially an ability to pick up dynamic interactions, is the idea that you would apply multiple of these at different sites and just see what emerges from it? Or, I mean, I'm just trying to think about like how you'd move forward, screening and determining what's real and not real based on the number of sites you have.
Yeah. So in terms of the kinetics, so we typically do our labeling for about 5 to 10 minutes. Mm-hmm. We're just now starting to do kinetic studies where we do things in very short times because the generation of the carbines is on the order of nanoseconds for them to react. So there's no way we could do something that short. But I think that some kinetic studies will be really interesting that way. And then in terms of identifying, the partners themselves, one of the things we want to do is to put the label on the partners and then look back at the complex. Yeah. Do they overlap? And that's something that we'd want to do.
Okay, yeah. Thanks.
Thank you.
I'd like to get a question in from our online audience, from Johnny Marvin. So why weren't EGFR or HER3 found to have interactions with HER2 by your new method?
Yeah, so we in some data sets we do see EGFR. It has to do with this cell line. It's not very highly expressing EGFR, which can complex with HER2, and same for HER3. So they just happen to be missing. Yeah.
Thank you.
Transmembrane residues get labeled using this technology.
Brain?
No. Residues that are in the membrane. So transmembrane spanning residues.
Right. So exactly. So when we look at that list of proteins, we do see some membrane proteins there because we think that the diazurine can go through the membrane on the other side. The resolution is about 120 angstrom. The membrane is 30. And some of those proteins are known to associate on the intracellular domain with HER2.
I guess what I'm wondering is the biotenphenols, for example, those you typically don't have a tyrosine in a transmembrane region. I'm wondering if the amino acid residues that are inside of the membrane also get labeled with this technique. Yeah,
Vamsi, almost all the labeling is on the extracellular domain of the proteins, but we do see some that are inside,
All right, we really need to move on.
Okay, yeah,
Thank you so much for your talk. All right, next up is Jiefu Li, who's going to tell us about in situ cell surface proteomics. Jiefu was previously at Stanford and is a new group leader here at Janelia and 40CP.
Good morning, everyone. I would like first to thank Jeannie, Jennifer, and Ron to put together this fantastic meeting. Thanks for attending in person or online. And I'm very glad to share with you our research on cell surface proteomics following Jim Wells, who is somewhat the hero in my field. I admire his work a lot, and also like to thank Jim for the really nice introduction to the importance of cell surface signaling and proteomics and the various methods we can use to profile it. And now I'm going to get into a very simple introduction to the tissue biology part. In the evolution of multicellular organisms, including ourselves, single cells assemble into the highly organized structures and coordinate their behaviors to achieve biological functions. To communicate with each other, to coordinate their behaviors, single cells need to talk with each other through signaling at the cellular interface. Thus, cell surface molecules and their signaling of central importance for the evolution, development, physiology, and pathogenesis of many multicellular systems. There are many ways for us to do the cell surface proteomic characterization profiling in culture cells, and that yield tons of new biological insights and therapeutic targets. But if we think about the tissue environment, it's a little bit more complicated. For any cell, its cell surface proteome is sensitive to and determined by many factors, including its cell type, its state at that time point, and its native tissue environment. As the gateway for mediating intracellular communications, cell surface proteome is often carefully regulated to serve the physiological function of that cell at that time point. To study tissue biology, it would be nice if we can find a way to profile cell surface proteome in native tissue environment while preserving all those critical features listed here. To that end, we collaborated with Alice Ting's lab, particularly her student Shuohan, to develop a method to profile the cell surface proteome in native tissue environment by using peroxidase-mediated biotination. In this method, HRP, the enzyme, is targeted to the extracellular phase of a cell. In the presence of hydrogen peroxide, it converts the substrate biotin phenol into phenoxyl radicals. These radicals can only diffuse within a short distance, although not that short, to the carbon chemistry, and then connected to nearby proteins. Biotinated proteins can then be captured and analyzed by mass spectrometry. Because we can control where to express HRP, this method enables the cell type specificity. By using membrane-impermeable substrate BXSP and the membrane-targeted enzyme HRP, it enables the cell surface specificity. This reaction only takes a few minutes to complete, thus allow high-temperature resolution. This paper was published 2 years ago, so if you are interested in the details of this method, please go to the paper. And next, I'm going to talk about one application in fruit fly olfactory development, and the other one is the extension to the mammalian system. So this is a collaboration with another graduate student in Li Qing's lab, Qi Xie. We use this method to study the transcription factor to cell surface executor to physiological function access. Transcription factors are often central commanders for cell fate and behavior, but often they reside in the nuclei without going to the surface, so they cannot mediate intercellular communications directly. Often they leverage cell surface proteins to do the job. Here is an example. In the developing fly olfactory system, this transcription factor called ACJ6 is a master regulator of the projection neuron PN lineage. It is expressed in half of the lineage, specifies their dendrite projection in the developing brain. But ACJ6 never goes to the membrane. Its wiring decisions must be executed by downstream cell surface proteins. Considering that it's a transcription factor, we originally thought that we just do RNA sequencing, then we'll find the downstreams. And turns out, among the 5 top regulated pathways by ACJ6, one is ER protein processing, and the other one is endocytosis. Thus, besides direct transcriptional regulation of cell surface proteins, ACJ6 can change the cell surface protein composition and dynamics through these indirect pathways, which could not be captured by RNA sequencing. We have to look at cell surface proteins directly. Thus, we profiled the cell surface proteome by comparing Y-type and ACJ6 mutant and found that indeed many molecules are regulated by ACJ6. And it's interesting to see that the mechanosensitive ion channel, piezo, is expressed on PN surface at development and is regulated by ACJ6. And we have heard several stories about piezo since its discovery about 10 years ago by Adam and his colleagues. This has been a star molecule in sensory neurobiology. But why would a central nervous system neuron express such a mechanosensitive ion channel at a developing stage? Could it do something related to development? So we experimentally tested it. In wild-type flies, these neurons shown in green do not target, innervate this area. And ACJ6 mutant, the transcription factor mutant, causes mistargeting, and piezo mutant phenocopied it. Importantly, when we supply back piezo in flies, it can rescue both piezo mutant phenotype as well as the ACJ6 mutant phenotype, indicate that piezo regulates dendrite targeting and it's a downstream of ACJ6. Now, the intriguing question is whether piezo does this through its canonical known function as a mechanosensitive ion channel. So we tested this by generating a mutant based on previous structure-functional studies. If we express Y-type piezo in cells, if we poke the cell, we can see a current. And in this mutant, it's completely silent. When we put this mutant piezo back to flies, we found that it can rescue the phenotype in both piezo mutant and ACJ6 mutant completely, no difference from the Y-type piezo, providing probably the first observation that this molecule can do something independently of being a mechanosensitive ion channel. And the piezo is only one example here. Our proteomic profiling allows us to examine many cell surface molecules, and we focused on the ones downregulated in ACJ6 mutant. And examined 11 PN types simultaneously in the brain. ACJ6 mutant causes mistargeting in all of types shown in red here. We then supply back each individual cell surface molecules to see whether they can rescue the ACJ6 mutant phenotype. If it's a complete rescue similar to Y-type, it is shown in white. If it's no rescue similar to ACJ6 mutant itself, it's red. If it's a partial rescue, then it's pink. When we supply back one specific isoform of ACJ6 itself, we can rescue most but not all phenotypes there. Taking piezo as an example again, supplying back it, we can partially rescue the phenotype in these 3 PN types but not the others. As shown in each row here, each molecule is frequently used by several PN types. And showing each column here, each PN type frequently uses a few distinct wiring molecules, forming a complex but very specific matrix here. This data immediately remind us of the combinatorial coding theory. Our genome encodes only about 20,000 genes, and only about 20% of them, as Jim mentioned, are cell surface molecules. But they need to specify more than billions or trillions of connections in our brain. If we use a one-to-one specifying system, we'll soon run out of molecule and could not complete the task at all. So the hypothesis is that these molecules can form combinatorial codes, which can significantly increase their coding capacity. Our proteomic data and genetic data allow us to experimentally test whether that's true. By simultaneously supplying back pairs of cell surface molecules, we indeed observed the broader and stronger phenotypic rescuing as shown by more white and light color squares here, providing a real-world data demonstrating the combinatorial coding theory. In this study, we mapped how a single transcription factor, ACJ6, leverages many cell surface molecules, particularly their combinatorial codes, to determine the architecture or targeting of many neuronal types. We hope that this pipeline method can be used in many other systems contextual questions to delineate the transcription factor to cell surface executor to physiological function axis. Now I'm moving to the mouse story. This is a collaboration with another graduate student in Lichun's lab, Andrew Schuster. We generated a transgenic mouse line to express cell surface HRP under the ubiquitous CAG promoter. It enables cell type specificity by using Cray lines, flip lines, or a combination of them. And this mouse line will be available at JAX very soon, so everyone can use it. And we found that this mouse line, this method, enables the cell surface biotination in many tissues and organs, as shown here by neutrality staining in green: kidney, heart, liver, fat, and intestine. we first applied it to the mouse cerebral Purkinje cells. We don't see any biotinylation shown by neutrality staining in green when we omit HRP or hydrogen peroxide. But when we run the reaction, you can see that the Purkinje cell surface, including its elaborated dendrite surface, are thoroughly biotinylated, but the intracellular compartments remain unlabeled. So you see this very typical whole structure there in neutrality staining. And we profiled the Purkinje cell surface at 2 distinct stages, postnatal 15 for a developing point and a postnatal 35 for a mature time point. And we obtained hundreds of proteins with partial overlapping. Geontology analysis of their localization found that these proteins are localized to membrane periphery, confirming the cell surface specificity of this method. And now we can, because we have developing and mature data set, we can compare the differentially expressed proteins as shown here in this volcano plot. And we grouped those proteins differentially expressed based on their molecular families. And you can see that the synaptic proteins, many of them get upregulated after maturation. And there is a distinct class of adhesion growth or guidance molecules. They are upregulated at the developing stage and goes down after maturation. While there is a different group of adhesion growth or guidance molecules, they get upregulated after maturation. Presumably, this group will be used for guiding development, and the other one may be used for structural maintenance or plasticity. This data inspired us whether we can use it to find new regulators controlling Purkinje cell development. And we focused on those unknown molecules shown here. And indeed, we found one called AMH4, which is a molecule that has never been studied in the nervous system at all. And we know that Purkinje cells grow these really beautiful arbors in wild-type animals. And when we knock out AMH4 by CRISPR, we can see that the arborization process is severely disrupted, much shorter dendrites and less complex. The interesting thing happened when we look at the overexpression of AMH4. We observed a very similar phenotype to the knockout, shorter dendrites and much less complex dendrite. Seems that the existence of AMH4 is not enough. You need to have it at a correct level. Then we asked whether the intracellular transduction of AMH4 is important for the function. By removing the whole intracellular domain from it and then overexpress, we found that then the phenotype is gone. It grows back to the normal size. And if you look at the HA staining, which is fused to AMH4, you can see that the Y-type and ICD knockout, they have very different localization. So when we zoom in, we can see that for the Y-type overexpression, they localize intracellularly from this puncta, and we have data showing that it colocalizes with indolysosome markers. And for the intracellular domain deletion, we can see that it's very surface localization, even within dendritic spines. Thus, that we know that there is a signaling motif, which is unknown so far, that controls the signaling transduction. And then seems that there is another function for the intracellular domain, is to control the localization. Then we looked at the intracellular domain and indeed found a 6 amino acid highly conserved endocytic motif there. And when we mutate this motif into anilines, we found that we got more presentation of AMH4 on the surface, but the phenotype, the dendritic arborization phenotype, comes back. So in this case, we now tease apart the localization signal intracellularly and the signaling transduction motif, which remain to be identified. More proteins goes to the surface, but the signaling transduction is still there. So you get even severe phenotype than the Y-type overexpression. Taken together, we developed a method to profile the cell surface proteome in native tissues with cell type specificity and temporal resolution. Applying it to neurodevelopment, we observed the global dynamics in development and how a transcription factor shaped the cell surface proteome, which help us to identify many novel and functionally important cell surface molecules for neurodevelopment. Combining the profiling with our in vivo functional studies, we observed the combinatorial cell surface codes for neuronal wiring and the endocytosis regulated cell surface signaling in dendrite arborization. with this, I'd like to thank everyone in the team, particularly my fantastic PhD advisor, Li Chun, for his support and guidance, Alice and her team for collaboration, and Steve Koss' team at Broad Institute for mass spec collaboration, and the Murthy group for the EFACE collaboration on the piezo project, and for Mark Davis for the project in immunology that I didn't have time to talk about. And before the Q&A, I would like to say that my lab now here is working on system cell surface biology by combining novel tool development and neuroimmune studies. And we have one open position for a cryo, ETEM, or chemical biology background postdoc. Please spread the word and join us. I'd like to take questions. Thank you.
For the in vivo, for the mouse studies that you're doing, I may have missed it, but are you giving the biotin substrate in vivo, or do you have to dissect the brain and then you label in a tissue culture dish?
That's a very good question. So for the mouse one, or for any thicker tissues, we have to dissect it out to do electrophysiological like sectioning. We originally worried about that could compromise the cell surface specificity, but based on the proteomic data, it turns out not the case. So the fly one, we did it in the intact fly brain because it's small, and for the mouse one, like the cerebellum, we did 300 micron sectioning, and the mass spec data was actually comparable to each other regarding the cell surface specificity or quality. Yes.
But can you give the substrate in vivo to a live mouse, or is that not possible?
That's probably pretty challenging because this labeling requires hydrogen peroxide. Although we use at a low concentration, it's still toxic. And there is a quenching step, which is really important for the specificity. And for that one, you probably need it to. We are now actually working on some new methods, try to push it in vivo. Hopefully, it can be successful. Yeah.
Yeah, I just had a question about the olfactory projection neurons that you said required piezo. What do they respond to? Do they respond to a specific subset of odors? And do you have altered behavioral responses to those odors?
Yes, so those are the projection neurons. They don't detect any odor response.
I know, but the glomeruli that they innervate.
Yeah, the glomeruli. I have actually not looked into that. It's a really good question. But the piezo expression in the olfactory PN seems pretty broad. Not very specific. Yeah.
Thanks.
Maybe we'll take Harry.
Hey, yeah, great talk. I'm wondering if you can speculate a bit about the idea of correlating this with a spatial map. So you could imagine if you have some highly stereotyped structure, say a nerve ring in a C. elegans, that you could, it's very stereotyped, right? I mean, could you imagine using your technique to give genetic precision to profile, some neurons and then correlate this with an electron microscopy map, say?
Exactly, that's a really good question. So Alice's paper with Kendall, they actually did like synaptic profiling, and there is another story I recently heard at the mass spec meeting that they fused to AIS axon initial segment, and they can profile the proteome there. So if you can use something to guide this HRP to a different compartment, you can do that. Alternatively, just fuse it with an antibody, and then you can guide to that nearby neighborhood and profile that area.
Jiefu, that was a great talk. I wondered, was there a biological theme to the proteins that changed in the volcano plots that you showed? In other words, why did those proteins change in their expression levels?
Yeah, so in flies, we observed something like the wiring guidance molecules, they basically, like a whole group, go down. And for the mouse cerebellum percanthus, we observed something different. So the guidance or adhesion molecules, one group was downregulated in maturation, and the other group upregulated. And the other thing I want to mention is that we always observe the protein processing enzymes like peptidases, proteases. They are highly abundant at the developing stage, and then they go down after maturation. This is highly consistent from the fly data to the mouse data. And the fly one, we actually, because genetics is robust, we actually did a screen and found quite a few novel peptidases or proteases that control neuronal development process.
Maybe one more question.
I'm really fascinated by independent function of piezo that you uncovered. And so do you think they might be in sort of axon guidance context? And so I'm wondering if you have any evidence to suggest they might be detecting guidance molecule directly, or kind of building on your combinatorial idea, do you think they might be acting as a co-receptor or scaffolding protein for other guidance molecule receptors?
Yeah, very interesting question. So far we have zero idea how piezo works there. It seems that because it's a super big complex, a trimer with like 600 kilodalton or something, and it's possible it has some co-receptors, ligands, and the other thing is that piezo seems can change the physical landscape, like the curvature of the surface. Then would that be possible, that how it works for neuronal wiring? That's something really interesting to explore, but we have zero idea.
Thank you.
Yeah. Thank you, everyone.
So to wrap up the session is Aubrey Weigel from Janelia. He's going to tell us about automatic organelle segmentation in volume electron microscopy.
Can you stop? All right. Well, it's really lovely to see everyone here today. A special thanks to the organizers, Ron and Jennifer and Janine, for the invitation to share the work of the project team here and for organizing this excellent symposium. Today, I will be telling you about the work that we're doing towards automated segmentation in volume electron microscopy. So, when we look at all the different organs and tissues in the body that we all, care about, and we zoom in, maybe a lot, what we'll come up with is just this collection of cells. And within those cells are the organelles. And as Jennifer pointed out yesterday, they're this important part in branching from what's happening inside the cell to the outside the cell to the whole tissue, the whole organ, and then the animal. And what we really want to know is how does that scaling work? And one way we can do that is by looking at the organelles themselves. So historically, light microscopy has brought us a long way in understanding these organelles and their functions. The beauty of fluorescence microscopy is it grants us protein specificity. And as Jennifer showed beautifully yesterday in her talk, it allows us to see organelle dynamics and how they interact with one another. And we get to see that with global context, right? We get to see all the organelles that we're labeling and throughout the entire cell or tissue. However, even with the multispectral approaches, we are limited by the protein labels. We only see what we label. And the resolution is maybe not optimal for some things. So on the contrast to that, electron microscopy gives us more global contrast, so we get to see more things. We get higher resolution, but with traditional methods, we're usually limited to a very small region of interest. So we lose all the context, and that is a problem as well. So an incredibly valuable tool that kind of selects the best of both worlds is focused ion beam scanning electron microscopy. Here we get near isotropic resolution, we get global contrast, and thanks to the ingenuity and the development in the Hess lab, we can do this across large volumes. So this is really exciting as a cell biologist going into tissue biology, right? So not only do we gain the resolution, but we retain the context now. Here are a few examples of some of these amazing large volume, high resolution data sets that are being produced here at Janelia by the Hess Lab and the FibSim Technology Group. These data sets are really rich with information, and it's just begging to be extracted. But because of their size, these are large, these are several terabytes in size. Because of their size and detail, extracting that information really is a mountain of a task in and of itself. So to start to attack this, the CoSIM project team worked to develop a machine learning pipeline to automatically segment organelles and cellular substructures within these data sets. We started at the beginning with a single cell, And we used existing data sets that the Hess lab had produced of conventional immortal cell lines to develop this proof of principle pipeline. And I'll walk you through what this pipeline looks like using a HeLa cell as an example. So we start here with the aligned EM data that was collected in the Hess lab. Then Grace and Allison in the group manually classified every voxel within a subblock of up to 37 different classes of substructures. So what were those substructures? What did we care about? Well, we tried to care about everything and be as agnostic as possible. So that included, your popular things and easier organelles like the mitochondria, the plasma membrane, to trickier organelles like the ER, the Golgi, lipid droplets, the endolysosomal system. We included cytoskeletal elements like microtubules, and nuclear components such as chromatin, the nucleolus, nuclear pores, and we even included ribosomes as a classifier. Then we put all that together, shown here are 15 such of those subblocks, and we just recently released a protocol on how to create these annotations yourself on Protocols.io, so you can check that out if you're interested for more information. And I'll also highlight that we used a different combination of classes for different machine learning setups. So we were interested if training everything together all at once or one thing at a time was better. And then I'll also just note out here in the bottom graph is how much training data we had from each data set. So to start out with, we had these 5 data sets, and the cell that I'm showing is HeLa2 there on the top, and it was less than a 1%, so it was only a fraction of a percent of the entire data set that was used for training. All right, so all that training data is put together, and it's used to train a machine learning classifier. We used a mostly standard setup with a 3D UNet architecture, as you can see there in the middle, and then this is trained repeatedly by feeding in blocks of raw data that produces some sort of output for the innermost part of the block, so the red part's there. And then this is compared to what it should be according to the ground truth annotations, and the parameters of the network are adjusted accordingly. And then this process is repeated hundreds of thousands of times in our case. And then from this, we can generate the predictions on the entire data set. So this is the exciting part. All these segmentations that we have aren't quite perfect yet. There are some things that we can do to help clean them up along the way, relatively straightforward stuff. So things like size filtering. If you have a large nucleus, there shouldn't be little bits floating around, so you can clean those up easily. Or applying a watershed algorithm to help over-splitting and over-merging. And in the case of microtubules, Nils from the Funky Lab was also able to improve the predictions quite a bit by applying a technique he developed that incorporates structural biases for the microtubules. So through this pipeline, we're able to get some nice segmentations for a number of organelles. And what's really exciting with these predictions is we can start to see the organelles in a way we've never been able to do before. And more excitingly, they're more than just a pretty picture, right? Now we can start to actually do something quantitatively as well, right? We can start to look at volume. We can look at the interaction between different organelles, their morphology at those interaction points. And you can find out more about these types of analyses in our recent publication, or you can explore it on our website. So with an ambition to democratize volume EM as broad as we can to the community, we importantly make all this work publicly available through our open access portal, Open Organelle. So here, together with the Hess Lab and the FibSim Technology Group and Scientific Computing here at Janelia, we provide a browsable, queryable interface for folks to explore all these data sets and the accompanying segmentations and find out more about the tools and such that we used. So on the site, you can filter the data sets to more easily find what you're looking for. You can browse pre-made views or customize your own view based on what segmentation you want to see. You can explore the data using NeuroGlancer, which is an in-browser viewer. You can also have 3D visualization capabilities within this viewer. You just double-click on a segmentation, and its 3D mesh will appear. On the measurements page, you can query all of the measurements that I was talking about there on the last slide. And you can look at things such as contact sites between 2 different organelles, and it will give you an interactable table of the volumes, the surface area, the contact IDs of those organelles. We also provide a glossary of sorts that contains how we defined the organelles for machine learning classification. And I'm showing here the centrosome, for example. And then you can also find instructions for navigating the website, other ways to visualize the data, including accessing it through Fiji and other tools if you do want to download the data. And we are actively inviting feedback on the site, so if there are tools or information that you're needing or wanting, we would absolutely love to hear from you. Okay, so all of this was within the COSM project team, and we developed this workflow towards automatically segmenting these organelles and other cellular substructures within the cell. But ultimately, what we wanted is to do this within tissue. So now within the CellMock project team, we're working towards this goal by leveraging the unique computational and technical expertise here at Janelia to support impactful research in interrogating high-resolution volume microscopy data. And of course, we're working to disseminate the resulting tools and techniques and data to the scientific community as well. So we're beginning by scaling our workflow to a couple different tissues in the mouse. So namely, we started with the kidney and liver. So we built our workflows with the scaling in mind, luckily. So that means that we didn't have to change too much to adjust for these increases in data size. You can see in this box just how much that means to increase. So the single cell is shown there in the little green box, and the chunk of tissues that I'm showing and as these two examples, which are 100 cubic micron boxes, are shown there for scale. And you can explore those more on Open Organelle as well. They're up and they're ready to be looked at. Okay, so what really helps to drive technology development is having some exciting scientific questions. So towards this end, what we did was to team up with Daniel Feliciano. His lab focuses on a phenomenon known as liver zonation. If we look at the liver, you can see it's structured as these hexagonal lobules, and it has a central vein, as you would guess by the name, at the center of the lobule, and then it has a portal vein out towards the corner of each of these hexagonal lobules. So the acinus is this territory from the central vein over to the portal vein. And it's really quite interesting. So while the cells are in theory the same all across that acinus, all of them are these hepatocytes lined up, they are experiencing a very different environment, such that the cells that are closer to the central vein are going to have a much different oxygen level than the cells next to the portal vein. And so this creates a very different metabolic environment. And as you could guess, maybe the organelles are also quite different. So this is very interesting and exciting to explore this difference across this gradient. And Daniel is specifically interested in the mitochondria and the ER interactions across this acinus. So shown here is a fluorescence image Daniel collected of a lobule with the mitochondria and actin labeled. And then just to relay this back to kind of the cartoon what the liver looks like, and you can see that the central vein is here in the center and the portal vein down in the corner there. And very excitingly, we now have a beast of a FibSim data set to accompany a fluorescent image like this, flowing from the central vein to the portal vein, so a strip of this acinus. And for the reference of size, again, so you can appreciate the scaling that's going on here, you can see the single cell data sets that I was showing at the beginning of the talk in the small green box at the top. Then the green box is that 100 cubic micron box that I was showing before. And now the whole liver acinus that we're excited to start working with Daniel on. And just a teaser of what this data looks like now, we're very excited to start mining this data set with Daniel, to start applying the tools that we developed in the single cell data sets and scaled it into tissue, and to use them now to help answer exciting biological questions, such as how the organelles change across the metabolic gradient of the liver acinus. So stay tuned for more in this arena. All right, so all of this work was really made possible by an incredible collaborative team effort, and really it would not be possible without the larger support of the administration, the labs, and the support teams here at Janelia. And if you didn't get a chance last night, I encourage you to go check out Allison, Caleb, and Will's posters. They're highlighted in green there, just outside the lobby, and you can talk to them and find out more about the tools that we're using and the segmentations that we're doing as well. And then I will join the crowd in encouraging folks to also spread the word that we are excited to have people come and join us. There's a lot of opportunities. We are eager to find more machine learning engineers. And you can find a list of those current jobs on hhmi.org/careers, or just reach out to any one of us with questions. All right, and with that, I have a whole minute and 20 seconds, so I guess extra time for questions. Thank you.
Great. I guess you're first.
Okay, great. That was a super exciting talk. I have lots of questions, but I'll only ask one.
Okay.
We can ask it.
When you were exploring, I think, that HeLa cell, did you discover any new organelles?
So the type of machine learning we were doing was not unsupervised, right? So it was supervised, so no. But as we personally go through the data and generate all this training data, there are a lot of opportunities to find things that you didn't realize or expect.
Right, perhaps not classified by, or well classified by the algorithm.
Right. And I would say that, in EM images, this usually falls in the category of, things like autophagosomes, the endosomal system, and tons of vesicles throughout the cell. So we don't find those pointedly, but you get to explore them more.
But do you have some sense as to what fraction of the voxels inside of a cell are unclassified and therefore potentially new organelles?
Yeah. Okay. So within the HeLa cell, I'd say that we have really covered a large part of it. And what's not classified is actually stuff that we know, so not as exciting. And it's the other cytoskeletal filaments, like actin and intermediate filaments, and we're exploring ways to, segment them more routinely. But yeah, I'd say like in the boring HeLa cell situation, there's nothing too exciting yet, but maybe when we get into tissue, there will be more. Yeah.
So the question is, does this critically depend on high pressure freezing cryo-EM, or could you do it with some sort of chemical fixation?
Yeah, so I can tell you anecdotally that our networks thankfully have been generalizable enough to not care too much. So all of the data sets that were within the COSIM work, those were high pressure frozen cells on a cover slip, and then freeze substituted and prepared for FibSem. But all of the tissue work, as you can imagine, is not, right? And our networks were able to translate over to them with some fine-tuning pretty straightforwardly. And we're working to constantly improve that. Yeah.
Okay. It's super, super cool. And I'm curious, when you train these models on, say, one data set, if you capture, say, another HeLa cell, how transferable is it? And similarly, is it transferable from HeLa cell to, say, a Jurkat cell?
Yeah. Okay, so within the first kind of project that I showed you, we actually had 5 different cells that we were looking at. They were all immortal cell lines, but it was 2 HeLa cells, a Jurkat, a macrophage, and a SUM159 cell. And even within the 2 HeLa cells, even though they were the same cell and actually from the same cover slip, so the same, things didn't transfer well between them all the time. And it really just depended on, like little imaging parameters or, contrast within that specific region. But we've been really stubborn about being agnostic and trying to include as much training data as possible from different data sets. And I think that has been helpful for us because now, we've now like tripled the amount of training data we have, and it now sees, stuff from not only just the cancer cell lines, but also, tons of different tissues, and things don't care as much now. And I think the reason is because of field of view. If you zoom in close to mitochondria, it's going to look pretty much the same across all these different cells, and that's really the strength of it.
Yeah,
This is truly exciting. Actually, follow-up question. I think you talk about the tissue of the cell heterogeneity. I want to know the organelle heterogeneity because, like for example, endolysosome system is very heterogeneous. Mitochondria, same thing. Like if you look at Crete, some have more, some. So are those heterogeneity included into the training? Yeah, how you deal with them?
Yes. So again, we try to. I mean, we don't do this in a really robust manner. But we're also not even close to the point of having too much training data. So we really just try to sample as many data sets as possible to access, to have as much heterogeneity as possible. Yeah. And that includes, like physiology and like staining, right? Yeah.
So something that got my attention was you're studying these portal triads, which have a very stereotyped geometry. And I'm wondering if you have tried registering these hexagonal units based on a portal triad. If you register them, in theory, you can superimpose all these units together, but the heterogeneity of the positions of all of these organelles should likely reflect the dynamics of how it fluctuates inside the cell. Have you studied that? Is that something you're interested in?
I mean, I think we're definitely interested in exploring it. The data set that I showed you here at the end just became available last week, actually Monday. So it's brand new, and that would be something really cool to look into, for sure.
Hi, great talk. I'm just kind of interested, and I don't know a whole lot about how FibSim works, but considering that all of this is sort of supervised by hand for the training data, is there any way to look at, say, like fluorescently labeled, like correlative light EM type stuff so that you have an easier sort of to collect training data set?
Yeah, so I didn't show this because this 15-minute time cap, but this is definitely something that we're exploring. The Hess lab and VetSeg lab have those amazing cryocorrelative FibSim and light microscopy setup, and it gives us a nice way to pair together like super resolution of both things, right? And so we're looking into how we can use light microscopy to add either more function or just help us get more training data. Yeah.
Thanks.
Great, thanks. I think that concludes this session in the morning. We have a break now, and we pick up again at 10:25, Janine? Yep. Okay. So it's about 20 minutes.
I like it.
we now have so much proof of concept that we can see the dramatic functional.
Hello, Celeste. This is Matt. I'm one of the techs here, and we have you ready to go on screen, and we'd like you to go ahead and share your presentation so that we can test that.
And then we can try to validate. That's what I'm thinking. Does that make sense? And then the other thing we can do is put that we've manipulated and characterized in vivo in these chimeric models. So we inject them in, and then you have basically we've characterized them after they've been back in the animal.
Have you seen Sergio Pascal's written paper?
Yeah, it's a little crazy. I mean, a little wild. She's always this beautiful work, but I'm just like, well, we next can have the mouse like talking and walking around.
So this project at Stanford, they generated this human frame organoid and implanted in rat habitats. And they integrated into the
organoid and put it into a rat.
What happened? And integrated into
it was just, having conversations and no, they did it with behavior. And not surprisingly, they got a little organoid in there. Yeah. And it wired into the brain. And they were trying to show it affects, they did a mutant, which is a wild type organoid that has like Timothy syndrome, so like autism. So, it was a lot of proof of concept. One issue if we treat this on these types of things. You have to keep doing more tech dev on organoids. But and what is it that you can really learn that you can't learn?
The biological insight.
Yeah, that's the part I'm not getting from any of this, and I think it could be that.
It's cool demonstration.
And they're not.
Hey, Celeste, that looks good. Do you have any videos or audio in your presentation?
So I just think like,
the environment is already aged for you to do something.
Yeah, or the vasculature, which is last year. So that's why we are using these chimeric models, because the genetics is clearly implicating the microglia. We know that these cells are relevant. We can see that it's affecting phenotypes. Now we want to know, how do we get them back into the mouse to see how they interact with surface. That's the so we can pull them back out again.
Celeste, do me a favor and say something. I just want to make sure we got program audio coming through today.
Sure, yeah. Can you hear me okay?
Okay, give me a second.
You can send your videos and post that here. Yeah, that's what you need for.
Yeah, that's right.